Hello everyone, welcome back to Heredity Biosciences YouTube channel. So previously we have uploaded a video on blood DNA isolation by using phenol-chloroform isoamyl alcohol. But today we'll do the blood DNA isolation by using Genes2Me blood DNA isolation kit. So let's get started. So first let's see what is inside of this Genes2Me blood DNA isolation kit. So first one is the collection tube, then mini column, then buffer BW2, buffer BW1, binding buffer, buffer LB, buffer AE, proteinase K, protease dissolved buffer. So first we will take a 1.5 ml fresh microcentrifuge tube. We labeled it. So first we will add 200 microliter of binding buffer. After that we will add 200 microliter of blood sample to the same tube. Then we will add 400 microliter of buffer LB. After that we will add 20 microliter of proteinase K to this tube. Then we mix it. After that, we will incubate the tube at 56 degrees Celsius for 10 to 15 minutes. I have already set the dry bath temperature at 56. So we will put this tube at dry bath for 15 minutes. Our incubation at 56 degrees Celsius for 15 minutes is over now. After that, we will add 100% chilled ethanol to this mixture. So here I am adding 200 microliter of chilled absolute ethanol to this mixture. Then we will mix it by vortexing for 10 seconds. After that, we will take a spin column. We label the spin column also. We'll transfer 650 microliter of this mixture to the spin column. Then we'll centrifuge it at 10,000 G for 1 minute. So now we'll discard the collection tube with the flow through. So we'll take a fresh collection tube, then we'll add buffer BW1 500 microliter. Then we will centrifuge it at 10,000 G for 1 minute. After centrifugation, we will discard the flow through. After that, we will add buffer BW2 500 microliter. Then we'll centrifuge it at 10,000 G for 1 minute. After centrifuge, we'll also discard the flow through. Then we will repeat the buffer BW2 step again. So I will add 500 microliter of BW2 again. Then we will centrifuge it at 10,000 G for 1 minute. After centrifuge, we will discard the flow through and we will do a dry spin at 15,000 rpm for 2 minutes. After dry spin, we will discard the collection tube. Discard the collection tube and place the column in a new 1.5 ml microcentrifuge tube. After that, we will add 50 degree warmed 40 microliter buffer AE. So here I am warmed the buffer AE at 50 degree. Now we will add it the center of the column. (40 microliter of 50 degree warmed buffer AE, adding to the center of the column. Now we'll incubate it at room temperature for 2 to 3 minutes. After 3 minutes of incubation, we will centrifuge it at 8000 G for 1 minute. After that, we will discard the column, and this is our DNA now. So now we have completed the blood DNA isolation by using Genes2Me blood DNA isolation kit. Thank you for watching. Please do like, share and subscribe our channel for more informative videos. If you have any questions or suggestions, please leave a comment in the comment box below. Thank you.